Thank you, Sharon. This is a pleasure to be here. I've so far really enjoyed myself. Unfortunately, the time has been a little bit short, and I've been a little bit busy, so I haven't seen much of Adelaide yet, but I'm looking forward to maybe a little bit this afternoon, and then I'm going to be traveling and visiting some of the other major cities in Australia, and hopefully I'll have some time in those visits to be able to see a part of your country. I think I scared Sharon this morning when I said that last night I decided that my talk needed to be revised, and so this morning I got up and I completely redid this. So what I can absolutely guarantee you, this is a talk nobody else has heard. Whoops. Okay, now wait a minute. All right. An outline of what I'm going to talk about. I'm going to cover 4 different general areas and try to give a perspective of how this impacts on clinical microbiology. And I'm going to, because it's an audience of rather diverse interest, I'm going to try to show how other aspects of what occurs in science can actually impact on diagnostics and patient care. So I've divided my presentation into 4 categories. The first would be a discussion of proteomics. The second, molecular diagnostics. Third would be lab automation, and the fourth, genomics. And hopefully this will be of interest to everyone. So we'll start off with proteomics. And the way we typically identify organisms historically have been by biochemical test or morphologic examination. And in reality, that hasn't changed for probably 100 years. So from my perspective, there is a great opportunity to be able to do things differently and better. Now, again, much of what I'm going to talk about is some work that I did while I was at the NIH. And so when I got there, I was there for 10 years. When I arrived at the NIH, it was by far what I think the best diagnostic laboratory in the United States. But they did things in a very traditional fashion. So what I looked at was opportunities to change that. The first thing we did is we introduced a sequencing program, starting with Sanger sequencing, evolving to Pyro sequencing. And we found that it was very accurate for identification of organisms, but it was time-consuming and expensive. We then moved to mass spectrometry, and specifically using MALDI-TOF mass spec. And what we found was that it was superior to both biochemical testing and sequencing, because in essence what you're doing is sequencing hundreds of genes and looking at those products. Now, the mass spectrometer, just to sort of put it in perspective, really consists of 3 functional units: a source of ion, a mass analyzer, and a detection device. The ion source ionizes the sample and transfers that to a gas phase, which is then going to be measured by the instrument. MALDI-TOF, or matrix-assisted laser desorption ionization, is considered soft ionization, and what that means is it doesn't destroy the proteins. So that actually in some work we did do is we were able to characterize the isolated proteins using this technology. The mass analyzer actually separates the proteins based on their mass and charge. The way this technology is used, the charge is generally 1, so you're really separating the proteins based on their size. And that separation occurs by passing the ionized proteins through a vacuum field where they're separated as they fly through space, or time of flight. The detection device is used to record the separated ions, and you get a profile that looks like this. Typically, for this type of mass spectrometry, you're measuring proteins that are from about 2,000 to 20,000 Daltons. You see a large number of protein peaks, but to actually identify the organism, there's probably only about a dozen that are actually used to do that identification. Subtyping actually takes advantage of the other protein peaks that are present. To perform this in the clinical laboratory is really very simple. And just as an overview, to prepare a sample of bacteria or yeast for this analysis takes about 1 minute. It's very fast. For molds, For molds and mycobacteria, it takes longer. I'll explain why in a second, but generally it's less than 1 hour, and then the profiles themselves can be generated in about 1, 2, 3 minutes. So the procedure is much faster than any procedure that was used before. You can take colonies from plates, you can take positive blood culture broths, you take those samples, you put them on a target plate, that's put into the instrument where it's analyzed, you get your profiles, and again, very rapid when this is done. Now, to prepare a sample, so there's some understanding of what's involved, is you touch the colony with a transfer device, very, very expensive transfer devices, they're called toothpicks. You then touch that to a target plate, you let that dry, you put 1 microliter of formic acid, and the formic acid will lyse the colonies, or the cells that are there. You then add 1 microliter of matrix, and you analyze the sample. So I think you can appreciate how simple it is to perform this. It's a bit more complicated for other types of specimens. So if you have a positive blood culture broth, what you have to do is remove the blood cells, the serum proteins, and hemoglobin. The way that's done is lysing the cells, concentrating the organisms, and washing the organisms. Again, a relatively simple process. For mycobacteria cultures, you need to kill the mycobacteria. We do that by heating the sample for 30 minutes at 95 degrees. You dispense the clumps by just physically breaking the clumps open. And then you lyse the cells using silica beads and formic acid in order to do that. So again, that process takes about an hour to do. But again, then the analysis is very rapid. And also for filamentous fungi, you need to kill the organisms. We actually used ethanol to do that. You break the hyphal structures, you extract the proteins, again, using formic acid, and do your analysis. This is a summary of all of the studies that we did in my laboratory with this technology. What we would do is build databases and then challenge the databases with clinical isolates. And what we found was the identification of aerobic bacteria, greater than 90% could be identified in the species level, with less than 1% resulting in misidentifications. For anaerobic bacteria, we built a comprehensive database. More than 75 species were included in that, and we were able to identify those organisms at the species level more than 90% of the time. For mycobacteria, we built a database with more than 80 species, and again, 95% of the organisms were identified at the species level. For yeast, we essentially took all yeast that had been associated with clinical infections, built databases for that, and then identified the organisms, and again, at 95% accuracy. Molds, and the mold work was done by actually one of the Australians we stole from Sharon's lab, a fellow that came to my laboratory at the NIH. Mold identification by this technology is a challenge because molds are identified morphologically, so that's really not a good gold standard to compare your MALDI-TOF with. So she took more than 150 species of molds, more than 400 isolates, and did genome sequencing to characterize the organisms. And then she challenged it with a very large collection of clinical isolates, and again, confirmed their identification by genome sequencing, and was able to demonstrate with that technology she could identify more than 95% of the isolates. And then finally, with blood culture isolates, greater than 80% could be identified directly from the positive blood cultures. The ones that could not be identified were because the numbers of cells were too few in the positive blood culture. When we compare the impact of MALDI-TOF identification with standard identification tests, this was a study that was done in one of the large US laboratories comparing MALDI-TOF versus standard identification. They looked at 952 isolates. MALDI-TOF was able to identify the organisms on average about 1.5 days faster than biochemical testing. And the impact of that was that it reduced the cost of organism identification by more than 50%. So very cost effective as well as rapid and accurate. Looking at the impact on antibiotic therapy, another study that was done, this was done in Texas, published last year. What they demonstrated was the direct identification of blood culture isolates reduced time to detection by, again, more than 24 hours. The proportion of patients that received appropriate antibiotic therapy was increased by 11%. Now that may seem low, but the reality was that their empiric therapy was very accurate. So despite that, they were still able to improve empiric therapy for patients by 11%, and that's without doing rapid susceptibility tests. So in another study that combined MALDI-TOF with rapid antimicrobial susceptibility test and introducing other technologies to be able to communicate the results rapidly to the physicians, when they compared their pre-intervention stage with the intervention stage, what they were able to demonstrate was there was a statistically significant improvement in being able to get patients off of inactive antibiotics, to decrease the time to an active drug, decrease the length of stay overall in the hospital as well as in the ICU, and the hospital costs were significantly decreased with this technology. So it demonstrates taking something out of the chemistry laboratory, applying it in the microbiology laboratory, and able to have a significant impact on patient care. And I think ultimately that's what we try to do with all of our diagnostic tests. So in summary for MALDI-TOF, all biochemical tests and morphologic identifications in my laboratory at the NIH were replaced with MALDI-TOF. And we were able to get the results for any of these organisms in less than 1 hour. We didn't require special processing for organisms like anaerobes and yeast. We were able to get definitive identification of molds and mycobacteria, again, the same day that the organisms were detected, rather than waiting days or weeks like we do traditionally. Blood culture identification was less than an hour after it was initially detected as positive. And we had faster, more accurate identification, which improved patient care and decreased hospital costs. So I think if I look at over almost a 40-year career, if I look at a dramatic impact by one technology, I think this is the technology that really has changed the way we do things in the clinical laboratory. Now let me move to a couple of other topics. Molecular diagnostics. Most of these instruments were in my molecular diagnostic lab at the NIH. We had instruments that were dedicated to do nucleic acid extraction, and other instruments that were dedicated to do amplification of the targets that we were looking at. There are other systems that integrate these two steps, large platforms. I give two examples. The BD Viper system and Hologic GenProbe has two instruments, Panther and Tigris. Essentially, it does what the previous slide showed, but it does it in a consolidated way. In recent years, there's been an introduction of smaller platforms and really self-contained platforms. We've gone from having PCR laboratories that involve multiple rooms where we separate processing so we can decrease contamination, to platforms like the BioFire Film Array and the Cepheid Gene Expert, where everything is contained in a disposable. So with BioFire, we have this pouch where you do very complex multiplex assays. And with Cepheid, we have these disposable cartridges that fit into the instrument that can be used for testing. I think these are remarkable systems. It has changed the way we do molecular diagnostics in the laboratory. I use both of these in my specimen processing area at the NIH, not in the molecular diagnostic lab, but the specimen processing area. The film array I was using for doing multiplex assays for respiratory pathogens, and the Cepheid I was using for screening for MRSA. Another approach is the BD Max system. This is slightly larger, but again, it could easily fit on the podium in front of me here. And the advantage of this system, in contrast to all of the systems I've shown before, is that it can be used to do processing of assays developed, in this case by Becton Dickinson or by one of our commercial partners. But in addition, it can be used to do processing of samples or tests that were developed in your own laboratory or in one of your colleagues' laboratories, and that's been migrated onto this platform. So when I refer to this as an, whoops, let me see if I can go back. When I refer to this as an open platform, I'm not referring to the fact that it can get contaminated. What I'm referring to is that anybody can put an assay on this platform. And so, in essence, it's the same concept of using the smartphone. Smartphones don't just make phone calls anymore. You have lots of different applications that you can purchase for that phone. Well, the same thing applies to this technology. You can purchase the MAX. You can use it for doing assays like MRSA or C. difficile assays that the company develops. You can use it to do assays that commercial partners develop. But more importantly, you can use it for assays that you yourself have developed. Now, let me give a couple of examples of how we were able to take advantage of this. And in my travels, again, I come in contact with issues that need to be addressed. And so in the first case, there was an outbreak of multi-drug resistant gram-negative bacteria. And so we developed a carbipinamase-directed assay. And in this case, it was directed against the three most common genes that encode carbipinamases in gram-negative bacteria, KPC, MDM1, and OXA48. We developed this multiplex assay and made it available for screening for colonization or infection with these organisms. The second example is an outbreak of meningococcal meningitis that's ongoing in Chile. And when I was visiting the head of the public health laboratory in Santiago, Chile, his complaint was that they didn't have an assay that they could use to confirm the diagnosis of meningococcal disease. So we developed a multiplex assay for him that detects Neisseria meningitis, Haemophilus influenza, and Streptococcus pneumoniae. That was done internally. We developed the assay. We took the assay and the MAX instrument, flew down to Santiago, Chile, trained the laboratory technologists there, did the validation work of the assay, and implemented the procedure. That process took 6 weeks. So very rapid, going from a need to a solution. Last month I was in the Middle East, and ongoing in the Middle East is an outbreak of a coronavirus, the MERS coronavirus, closely related to the SARS coronavirus. And the need there was for a diagnostic test, a safe diagnostic test. One of our clinical partners developed a MERS assay. And I requested that before I left for the Middle East trip. 2 weeks later, the assay was available so that they could use this. So again, demonstrating the speed at which assays can be developed for a platform like this. Similar work has been done for H7N9 influenza, which is currently causing an outbreak in China. And the concern, obviously, is that that could become a pandemic strain. So in each one of these examples, we developed an assay, a multiplex assay, in some cases a very complex one, for this type of a platform. So I think it's going to change the way we do molecular diagnostics in the future. Becton Dickinson was the first to develop this open type platform and make it readily available and easy to use by literally anybody that could use that. I'm absolutely convinced that we will see many more of these types of assays in the future. And I think that's how we're going to finally be able to take molecular diagnostics and make it practical for clinical laboratories to use that. Laboratory automation. I talked a little bit last night about laboratory automation, so I apologize to those that heard part of this. If we look at today, we've been able to automate a number of processes in the laboratory for doing diagnostic testing. So you have blood culture instruments, you have identification and susceptibility testing instruments, you have the MALDI-TOF to do identification, and you have molecular diagnostics. But the problem is that we really haven't totally automated the laboratory. And if you look at other industries, the auto industry is, I think, really the best example. We've gone through really custom hand-built cars to the ability to use robotics to build cars that are highly sophisticated and much more reliable than what we saw in the past. We also have to just look around in our colleagues in other laboratories and see that automation has been introduced in chemistry and hematology, immunology for a number of years. And so the question is, why not in microbiology? Well, we actually have much more complexity than what you would see in the other laboratories. And just think about the types of specimens. Rather than just bloods and urines, we have all different types of tissues, different sizes, different volumes, and complex testing requirements. And so really it has been a challenge to develop automation in these laboratories. I think what becomes critical is we have to start thinking differently. If we're going to make changes, if we're going to automate the microbiology laboratory, we have to think about this a little bit different than what we've done historically. Now, this is an example of Cambridge Public Health Laboratory in London 25 years ago. And what I'd like you to notice, you know, they have Bunsen burners, they have ampoule containers or disinfecting containers, incubators. See those chairs? Remember those chairs? They have all sorts of different things. Well, if you look 13 years later, they got the same stuff. They haven't made any changes at all. Maybe they've painted the walls, but really it's the same thing. I think they have the same technologist there, he's just lost a little bit more hair. If you look 9 years later, everything was removed. And the next month, this was introduced. So this is one of the first laboratories that really was totally automated in the world. And really, it's a remarkable process. So as we look at automation today, and this is BD Kiestra, which has really been the leader in this area, there's all different levels of automation. There's automation for the benchtop, where you automate a very specific function so that you can do that reproducibly and do it well. You can do what's referred to as work cell automation, which really is a self-contained microbiology laboratory for relatively low volumes, up to about 600 specimens a day. And then you can do total lab automation that really can accommodate any size specimen volume that the laboratory is going to deal with. As you look at the components of automation, one of the earliest has been automating the initial specimen processing, where you have devices that can streak the plate. So as an example, the plate here, you can notice there's a metal bead that's in the center of that. Well, that bead is moved across the surface of the plate to streak the specimen on the plate, and it's done by a magnet underneath. And so, as you can imagine, you can change that streaking pattern in any way that you want, including, I guess if you're so inclined, you can have the technologist initial the plate when they get done. But what this allows you to do is it reduces the processing errors, it improves the recovery of organisms because you have well-isolated colonies, decreases the technologist time because they're not doing this, the instruments are doing these types of functions, and it improves safety because you're not exposed to aerosols that are created by our traditional procedures. As you move into other types of automation, those plates then can be taken by tracks into a intelligent incubator, and this incubator then eliminates the delays that we see today in moving a specimen from where you do the initial processing to the incubator, or maybe more importantly, the plates stay in the incubator rather than after a certain period of time taken to a workbench where the technologist puts them there and lets them sit there for hours while they do work. This type of a system allows examination of the plate at user-defined intervals, much the same way that the current blood culture and identification systems look to see, is there growth in this blood culture bottle? Or can we examine the susceptibility testing plate? In the same way, these types of systems automatically examine the plates and tell you whether there's sufficient growth that you can then use to identify the organisms and do the other diagnostic tests. And it also allows you to rapidly retrieve the plates. This shows the, shows the arm that's grabbed one of the plates. And what I would encourage you to do is stop later this morning by the BD booth, and you actually look at these incubators and see them in action. It's really quite different from what we've done historically in the laboratory. And then you can do digital plate reading. And I think this is the real strength of automation. because what it allows you to do is look at all of the plates from a patient at one time and be able to compare those and say, you know, what is the growth, you know, what did the specimen look like last week that we received on that patient? Is it the same organism that's on this plate? Much like with your iPad where you can increase the magnification to look at the script or a picture on an iPad, you can do the same thing with this and look at the plates in greater detail than what's available naturally. And then the digital images can be taken at set intervals so you can see the progression of growth over time. And finally, the images can be reviewed remotely, either in the laboratory, in a reading room, at home, or thousands of miles away. Much like today, in many hospitals, the radiographic images are not read in the hospital, but it's read distantly by somebody that just sits in front of a screen and does that type of analysis. We can do the exact same thing here. This also gives you tools so that you can do a better job managing the laboratory. So you can have large monitors that display a summary of all the cultures that have been processed and read by a technologist. So you can look and say which technologist is efficient, which one is not, and be able to assess that really very objectively. You can organize the workflow for real-time workflow management. Right now what we do in a laboratory is the work sort of is done when we're ready to do it. In this case, the work should be done when the testing is ready to be examined, and that's really the difference. And that allows you then to schedule your staff according to the workload requirements. All right, so that's automation. That's today. That's the way work is being done today, and I think that's really what's very remarkable. I'd like to move to the last area, and that's genomics. And what I'd like to do is to show how the work that was done in the Human Genome Project and the Human Microbiome Project can apply in the clinical laboratory. So if we look at the Human Genome Project, this was a 13-year international effort that was initiated in 1990, concluded in 2003, that was funded through the NIH and a number of other organizations, both in the US and internationally. The goals of this project was to determine the sequence, roughly 3 billion nucleotides, that compose human DNA. So we're going to say, what is the sequence of the whole human genome? The goal was to identify the protein encoding genes. There's about 20 to 25,000 genes in human DNA. To store that information in accessible databases so that other people would be able to have access to that. To improve tools for sequence analysis and transfer related technologies to the private sector. And to address the ethical, legal, and social issues that come from this type of work. In other words, can you discriminate against a person that's carrying a gene that encodes for, let's say, breast cancer? Well, they dealt with these issues at that time. The interesting, I think, comparison of this type of work is much like sending a man to the moon. And you can question whether there's value in sending a man to the moon, have him stand there, hit a golf ball, and come home. But one of the things that came from all of that work was technologies that we use today that really has shaped our lives. The same thing occurred with the Human Genome Project. Many of the technologies that were developed to accomplish this goal really impact directly on diagnostics today. When that work was completed, they then turned to a different target, and that's the microbial population that lives in and on us. To step back and look at this microbial world, we're outnumbered 10 to 1. For every human cell in our body, there's 10 microbes. So we're really more of a bacterium than a human. If you look at a 200-pound adult, they're walking around with about 2 to 6 pounds of bacteria. So I guess if you really wanted to go on a diet, you get rid of the bacteria and at least lose a few pounds. Not advised, as you see in a second. The human genome has 22,000 protein genes. The human genome, the human microbiome, has 8 million unique protein genes. So there's a lot more metabolic potential in the microbiome than there is in the human genome. And the last point I want to make is our relationship with the bacteria that we live with. And most of the microbes that we have on our body surface are bacteria. This study actually looked at bacteria, yeast, and viruses, but the main focus was in bacteria. But our relationship with bacteria really is a beneficial one. Either the bacteria and the host are benefit, and that's mutualism, or maybe the bacteria aren't benefited, but the host is, and that's commensalism. So we take advantage of those bacteria to do lots of metabolic processes, to stimulate the immune system and maintain the integrity of the immune system. We have tended historically to look at bacteria as being bad, and the reality that really doesn't occur. Parasitism is pretty uncommon, and that is where the bacteria benefit, but we become sick. And competition really isn't a good thing for either, because they both lose. And you can imagine if a bacterium infects a person and kills the person, well, the bacterium doesn't have a host anymore. And so both lose in that case. But when we look at parasitism and competition, it's relatively uncommon, and I think that's an important point to recognize. So the human microbiome was really a project that was initiated after the genome work was completed, taking advantage of the technologies that were developed for the genome. This was a 5-year study that was just completed last year, and what they focused on was looking at the microbes that colonize healthy human adults. And I was involved in the initial studies when this was being planned, and there was lots of discussion, you know, how do we select the individuals, what sites do we select, and I think in retrospect, much of that discussion turned out to be unnecessary because of the conclusions we were able to pull from the way the study was organized. It was funded through a $153 million Common Fund NIH grant, as well as some of the individual NIH institutes also provided additional money for this study. The goals were very similar to the Human Genome Project, to develop a reference set of microbial genomes, to characterize the human microbiome, develop new tools and technology to perform this task and analyze the data, to establish a resource repository so that this would be available to any investigator that was interested in this, to elucidate the relationship between the disease and the microbiome changes. This is really work that's ongoing now. And again, to address ethical, legal, and social implications of this work. The way the project was organized, the samples were collected from a total of 242 healthy volunteers. Samples were collected at 2 sites in the United States. 5 sites were sampled. You can imagine, you can sample all sorts of sites, but it was really decided to be very, very specific about the sites that were going to be examined and how they would collect those samples. These included 9 oral samples, the nares, the stool specimen, 4 skin samples, and 3 vagina samples. Specimens were collected at multiple time points so you could look at the changes in the microbiome in these healthy individuals over time. And 2 types of sequencing were performed. 16S ribosomal RNA sequencing, which was used to define the numbers of organisms that were present in individual species, and then shotgun sequencing, which sequenced the entire genome, and that was used to be able to define the genes that were present, the protein encoding genes that were present in the genome sample. And as I said before, the vast majority of organisms were bacteria. Now, this rather complicated slide, let me walk you through it because it does illustrate a point I just made. So we start off with a sample that was collected. Let's say this is a scraping of the gingiva in the inside of the mouth. DNA was extracted and subjected to 16S sequencing, and what that would do would be able to define the number of different types of species that were present and the relative proportion of those species. So it really defined the composition of the population that was present in that sample. DNA was also extracted and subjected to shotgun sequencing, which really sequences genes along the entire genome. And what this ends up doing is telling us what is the metabolic potential, what are the genes that are present in that sample, irregardless of the identification of the source of the genes, what genes are present there. So we get two very different bits of information by these two types of sequencing. Now, I will say the shotgun sequencing was not done for all samples because it's a tremendous amount of genetic material that's generated from that. But as we'll see, it wasn't necessary to do additional sequencing. As we look at the population of organisms on the body, what we see is there's substantial variation in the number of species and the types of genes that are present at each body site. So the number of species are what's in red, and what's in blue is the number of genes that are present. And you can see, looking at different body sites, there's really quite a diverse population that's present. If we look at what's referred to as the richness at each body site, and what this really is you can consider this data representing the number of species that were present. The very tall bar here, I know it's difficult to read here, but this is the stool specimens that were collected. Ignore the bars here for this purpose. But there is roughly 40,000 species that are present in the stool, different unique species. If you look at the mouth, there was about half that, or about 20,000. And then the nares, skin surface, and the vagina, there is between 5,000 and 10,000. So lots of different species that are present in those samples. If you look at the number of species that were common to at least 95% of the individuals that were sampled, and this is the core microbiome, it's a very different story. So we'll go back to stool just as an example. This is the core microbiome, and again, two different sequencing techniques that were used, 16S sequencing targets that were used, and that's why we have the blue and the yellow bars, but we're going to see roughly the same results. Somewhere between 5 and 10 species were common to everybody. That was surprising to me because I thought the core microbiome was much larger than that. If you look at the oral cavity, this is where you see the most core organisms or organisms shared by all individuals, but still it's only between 20 and 25 people. Skin and the vagina, the nares, again, less than 5 were found there. So what it tells us is the core microbiome is very small. So we're each very unique as we think about this. And if you look at, this is another one of the studies that looked at the core microbiome from a number of different samples: hair, nostril, skin, vagina, oral cavity, esophagus, the stomach with and without Helicobacter pylori, and the colon. Consider that the colors represent different groups of bacteria. And so just visually, you can see that in the vagina, you have one predominant group of organisms that really is not very common in the hair. And you have a group of organisms in the hair that's not found really in any of the other samples that were looked at. What this means is that at the individual sites, there is a common group of organisms, a common family of organisms, if you will, but the individual species within that family can vary tremendously. Another way of looking at this is that what we see is that in each one of these sites, you have a common metabolic potential that's in that site, or a common pool of genes that all provide the same function. And that function can be unique for that site and different from other sites as you look at it. Another way of looking at this is looking at the skin microbiome. It's 4 different sites, and I've circled each site. We have the antecubital fossa, or the inside of the arm, the back, in this case the nares, and the heel. We have 4 individuals in each one of these groups, and what you see is, based just looking at color, first of all, the color here is obviously very different from this, and very different from this and this, which means that the general population of organisms is different from each of these different sites, and it's different between different individuals. And so this really emphasizes again that point that I made. The core microbiome differs dramatically, but I'm not showing the metabolic functions, but the metabolic function at each of those sites is common. That becomes very important because as we think about disease isn't necessarily a change in the bacteria that are at a body site, but rather a change in the metabolic potential at that body site. So this is a study that looked at a large number of women and looked at the population of the vagina. The first two columns represent clinical criteria defining health and vaginitis. And for our purposes here, if it's green, it's from an individual that's healthy, and if it's red, it's from an individual that has bacterial vaginitis. If you look down here, this is primarily the healthy ones, and what you see is that most of the bacteria in these women are the same organism, or at least from the same family. So as an example, they'd be lactobacilli. The lactobacilli are able to ferment glycogen producing lactic acid, and lactic acid is necessary to maintain a healthy vagina. And so you have a population of organisms, whether it's lactobacilli or a related group that's able to produce lactic acid, that is able to maintain health. In contrast, as you look at individuals that have disease, you see in this case a very complex flora, and I can tell you what's happened is they've lost the function to be able to maintain a healthy pH in the vagina. So what we see here is two different microbial populations, one representing health and one representing disease. And you can imagine that we can predict disease not by looking at this, because you already have diseased patient, but looking at a progression from health to disease and actually prevent disease. So as you look at diagnostics, I think in the future we'll be preventing disease based on these types of studies and not trying to do diagnosis like we do traditionally. So what's the impact of the microbiome research on the clinical microbiology laboratory? I don't think we're going to replace microbial identification using sequencing to replace MALDI-TOF as an example. I think MALDI-TOF is going to have a very important function in the clinical laboratories. We may change the type of mass spectrometry we do to have it more sensitive, but this is rapid and accurate, and so I don't see microbiome work changing this. What it will change, though, is that we can start predicting the composition of populations of organisms, not individual organisms. We can predict virulence. I didn't show you some of the studies that look at that, but we can characterize which Pseudomonas is actually going to cause disease in a patient rather than just saying the patient has a Pseudomonas. And we can use this for typing. And again, this afternoon I'll present some additional data on how we use this type of data for typing and doing epidemiologic investigations. I think we'll see new approaches to diagnostics, what I referred to before, where we'll predict disease rather than predict the susceptibility to disease rather than diagnose disease. And I think, to put it in perspective, there are technical challenges with this. This is very complex work today, very expensive, and really not done in very many institutions. I was fortunate to be involved in this work when I was at the NIH, but there's not many places that can do that. The pre-analytical processing, the sequencing, the data analysis is daunting. But just like sending the man to the moon, just like sequencing the human genome, technologies will be developed that will allow this to become, I think, routine diagnostics in the clinical laboratory. Maybe not in the next 5 years, but clearly between 5 and 10 years, we're going to see that occur. So as I look at the future of microbiology, I see lots of changes ahead. I think we are in a period right now that really is unique because we've seen the introduction of changes that really were unimaginable even 10 years ago. And I'm very optimistic that over the next 10 years, we're going to see dramatic changes in the way we do things, and I think it's going to have dramatic impact on the way we take care of patients. So with that, let me end, and thank you for your attention.